खिलाने के लिए उसके पास इंतजाम है। मैं इतनी लंबा सफर क्यों कर रहा हूँ, मैं अपने घर में बैठूं, यही मुझे खाना मिल जाएगा। देखिए, ये बात उन्होंने कही थी। इब्राहिम ने जो जवाब दिया वो आप सबके और हम सबके सुनने के लायक है और बड़ा सबक है। कहने लगे, शफीक तुम बीमार चिड़िया की तरह क्यों बने? तुमने अपने आप को उस सेहत वाली चिड़िया की तरह क्यों नहीं बनाया जो दूसरों को खिलाने का जरिया बन रही है, दूसरे के मुंह में दाना डाल रही है? तुम वैसे क्यों नहीं बने? बात एक ही है, लेकिन दो अलग अलग जावियों से और दो अलग अलग एंगल से शफीक ने देखा उसी चिड़िया को एक दूसरे एंगल से, उसमें उनको कुछ दूसरी चीज नजर आई। इब्राहिम ने उसी चीज को दूसरे एंगल से देखा, उन्होंने उससे एक एक एक, एक अच्छा सबक निकाल लिया। यही है आज के लिए, खुदा ने हम सबको दिमाग दिया है, हम सबको इल्म दिया है, और हम सब अपने अपनी नॉलेज का, अपने अपने विजडम का, अपनी जो उसकी दी हुई नेमतें हैं, जो उसके गिफ्ट्स हैं हमारे पास, उसका इस्तेमाल हम दूसरों के लिए करें। इसी में हम इंसानियत की भलाई है और इसी में हम सब का फायदा है। मुझे लगा कि शायद ये मेरी बातें आज की आपकी इस कॉन्फ्रेंस के लिए फायदेमंद होंगी, इसलिए बच्चों के सामने मैंने ये बात करी।
Shukriya, Imam Saab. I would now request Professor S. Raisuddin, Advisor Research and Head Medical Elementology and Toxicology Department, School of Chemical and Life Sciences, for the welcome address to the August meeting.
Assalamu alaikum. Good morning to all. Honorable Vice Chancellor on the dais, Professor Dr. Subrata Sinha, a very renowned neurologist and neurochemist. Dr. Prakash Babu, the official of the Neurochemistry Society for Neurochemistry India. I welcome, sir, all of you on this very important program, which is, I think, second in the consequence. I think last we organized a different kind of SNCI program, and today we are assembled here with discussing on the aspects of neurological legacy of neurological disorders. There will be a brainstorming session, as well as the participants and delegates will discuss about different approaches. By that, these neurological disorders can be tackled. So I am happy to see a very compact but very elaborate at the same time program has been prepared by the organizing committee. Especially, I must congratulate Professor Dr. Sohail Parvez for making such a program where young delegates as well as the mature delegates will have a kind of a meeting point, interface, interaction, as well as discussion on varied topics of neurological disorders. So there is nothing much to elaborate on the neurological disorders. Every day we see some kind of new terminology for neurological or rare diseases. I think a couple of days, maybe day before yesterday, we were reading the interview of Nasiruddin Shah, very celebrated and very decorated actors of Indian cinema as well as theatre, that he was suffering from a disease, monomytomania. It's a new kind of term. I also heard this disease first time that he is suffering, and that is related to fixation with certain kind of phrase, certain kind of words. He said he could not sleep. When he is sleeping, he is always haunted by a kind of phrase, a kind of word. So neurology and neurological disorders are new types of. Many films have been also produced on such kind of disorder. I think My Name is Khan, as well as Pa. And so this is kind of very, and many times even just recently released films have that highlight the neurological issues. So research is very good. Research is very strong in our India in this field, and Jamia Hamdard is one of the very vibrant center where neurological disorder and neurotoxicological, neuropharmacological, neuromodulation research is going on, including our own department, where this subject is flourishing like anything and getting support from all major funding agencies like SERV, ICMR, TEST, and so many other funding agencies. And there are three laboratories, including Dr. Sohail. My lab is also has some kind of different neurological research in pharmacology department and also in other departments. Even pharmacognosy is also trying to find out some plant extracts, and even Center for Translational Clinical Research also has neurological. So neurology is catchword in our institution, and very rightly the the Society for Neurochemistry India has chosen this center as one of their leading centers. Society is very old. I don't want to highlight. It's a 43 years old society. I am right. I guess that was founded in 79. So such a long society having very eminent people associated with it. Dr. Sita, Dr. PK, and so many people are there. So I hope that during your participation for this one day's program, you will listen from many renowned speakers, including Professor Sinha and Professor Dr. Prakash Babu, and some young speakers which have been lined up in this program. That includes people from even funding agency speakers are also there, including from ICMR and Science and Engineering Research Board. Their funders, those who handle the projects, they will also guide you at how we can take up. So neurology, even in our Ayush Ministry, CCRM is also having lots of program for neurology. So with these words, I welcome all, including on the dais, all teachers, all faculty members, all delegates from other institutions to this one-day deliberations organized by the Society for Neurochemistry India department on the department of in collaboration with Department of Medical Hematology and Toxicology and other departments, pharmacology, pharmaceutical chemistry, and other departments of the department. So I hope that you will love and enjoy these deliberations and another academic things. Visit the lab of Dr. Sohail and our department laboratories. So again, I welcome you all. Thank you very much. Have a nice day and all deliberations today. Thank you very much.
Thank you, sir. We are fortunate to have on stage Professor P. Prakash Babu, Secretary General, SNCI. He is an eminent researcher and educationist at the University of Hyderabad, having done his PhD from University of Hyderabad, followed by 2 postdocs from Japan and UK. His main interest areas of research include cerebral ischemia or ischemic stroke, cerebral malaria, glioma, meningioma, and epilepsy. Sir has guided more than 20 doctoral students. He has published more than 100 research articles and presented at more than 150 national and international conferences. He has served as Pro VC, University of Hyderabad, and is currently serving as Dean in Charge, School of Medical Sciences. I would now request Prakash Babu, Sir, Secretary, SNCI, for the inaugural address.
Thank you for inviting. Respected Vice Chancellor, Jamia Hamdad, Professor Afshar Alam, Professor Raisuddin, my friend and a dynamic leader, Suhail, and the eminent scientist, teacher, and a mentor, Professor Subrata, my colleagues of the dais, faculty members, students, and other supporting staff. Very good morning, all of you. It's my pleasure and privilege to be here as part of this SNCI. As earlier people, they have been indicating about the society and its past history. I'm not going to touch that all these things, but I would like to impress upon the eminent personalities here. So this society is majorly targeting the students at the young age, catch them at young, so that the students can be lured towards this amazing research in the brain. And as you know, our logo indicates that the brains are gathered to know how brain works. So this is our logo actually. So the society, we organizes annual meetings. Apart from that, we organize workshops. Workshops we majorly concentrate to train the young minds, to give them a hands-on experience and hands-on training to the students. during this, and also we have apart from the society, we have the local chapters. These local chapters indicates that there are around 10, 12 local chapters, and one among them is very active Delhi chapter, for which the Professor Sohail is actually coordinating. And we have the local chapter, he is organizing this meeting. So the local chapters also, they promote the research and training to the young minds especially. So during the year, so we organize several places, the local chapter meetings, wherein specially train the young people. So we have one in here, Hyderabad, Chennai, Bangalore, Northeast University, Gwalior. So there are several places. We train the students. So in between, throughout the year, some activity or the other we will be doing it. And IBRO and the International Society for Neurochemistry or APSN, these are the people that they are closely working with us. So therefore, this is the mandate that with which we have been taking up. So this meeting with Professor Sohail is a very dynamic and a important one of the pillars of the society here we have. So the institute, I also appreciate the institute and the vice chancellor, subsequent vice chancellors also. They have been cooperating, and we are feeling that this is another place, like a headquarters, this is another headquarters for us for the society. So this is what a kind of feeling that we have been given, because the faculty members are so enthusiastic to organize this. And Professor Sohail especially, his lab is having so many neurological diseases models. Not many labs are having. So having models, animal models, is very important to design the experiment. For every disease you will have, but not the neuroscience. It's very difficult to have the model in the neuroscience because it's very difficult thing. And Professor Sohail is having another important thing is he's also keep open his lab for the young minds to train. Not many labs will open the lab for this. So this is what I really appreciate Professor Sohail and his team of scholars and also colleagues, faculty members. Actually, there are many colleagues are here. They are with him. So team building is an important, and they have very good team to organize such a kind of meetings. Really, I feel really proud to have him as one of the organizers of the SNCI, and even we would like to give him a more responsibility on behalf of the society in years to come. So, and apart from that, the local chapter, this also is another important milestone in this meeting that I would like to announce, that we have even members also in the US or outside the world, outside India. And this time, there are more than 10 people are got into society, majorly from the Jamia, students of the Jamia, those who went from abroad, and they are faculty members and there are postdocs. There are, so with all this possible because of the Professor Sohail and his team. And I'm announcing that one, we have the local chapter, means US chapter of society. Today, I'm proud to announce that on behalf of this Jamia Hamdard and this meeting, we have that one. And again, so the major contribution is the Jamia Hamdard. So with this actually, so there is no actually much time as the Sinha has to go and attend. So I really congratulate the Professor Sohail having this kind of meeting. It's a neurochemical legacy of the neuro. So this is the actually very important area. Not many people are addressing any neurological disease. Though we have been working for the neurodegeneration and all, but the neurological, so neurochemical, so neurotransmitters, they play an important role in any disease. So that is not many people are addressing, and this meeting is going to cover many aspects of that. And also students are having so many posters and encouraging the students to come and present the research work and all. So with this, I congratulate the Professor Sohail and his team, and also I thank the team, and also I thank especially the university for providing the facility and making this as another important place for the society. I thank the Vice Chancellor for giving this opportunity and helping us. So thank you all. I wish you good luck.
Thank you, sir. We have the privilege of having amongst us today our Honorable Chief Guest, Dr. Subrata Sinha, sir, who is a perfect blend of simplicity, sincerity, and efficiency. The extraordinary personality, a nobleman, a prophecy of distinctness. He has done his MBBS and MD Biochemistry from AIIMS, New Delhi. He completed his PhD from MRC Toxicology Unit, Cashilton, and the Chester Beauty Institute of Cancer Research, London. Sir has insightful experience of working with research institutes, heading the Department of Biochemistry, AIIMS. He has also served as director at MBRC. Sir has a multidisciplinary collaborative research program at AIIMS on the genetics of glioma. Another area of research relates to recombinant technology. have also been working on neurodevelopmental of the genetics and molecular processes associated with hereditary learning ability by studying 3 generational families. These have helped identify distinct dyslexia and also some novel neurodevelopmental pathways. He has been awarded with the Swarnajayanti Fellowship by DST. He has been an elected fellow of the INSA, IASC. He is a fellow and was president, Indian Academy of Neurosciences. I would now request Honorable Chief Guest Dr. Subrata Sinha sir for his address.
Yeah. Good morning, everybody. And you know, this hall is very familiar to me. I've come here very often, and I have interacted with people here. And first of all, I must give my thanks to Professor Alam, the Honorable Vice Chancellor, Professor Raisuddin, Professor Prakash Babu, and Professor Suhail, always Suhail, for having me here. And it's one place where I feel really very welcome and feel at home. Uh, First of all, SNCI is a society which has been carrying the banner of neurological or research on neurobiology applied and basic for decades now. And it is a big, it's a standard bearer of this discipline, I must say. And I am very happy to know that the Delhi chapter is so active and Israel is so active, and they are, providing a dynamic boost to the subject. Okay. Before this, I would also, before we move on, just I'll briefly like to also talk about recently one of our colleagues in biochemistry department, Professor Rajeshwari. Suddenly, her sudden demise really saddened us all. And I would like to remember her and her association with SNCI when I'm giving this talk. Having said this, I thought that, I am not a good speaker and I don't really, so I need something specific to talk about. And therefore, I felt that I would really talk about our work on some, not more than our work, but to give you a perspective on how the world of tumor biology is changing. I realize it's a bit removed from neurochemistry in the sense that we are not talking of any neurotransmitters, but we are talking of molecular signaling in tumors. Now, one of the reasons why things have applied biology, let me say, has changed is that there is clinically today, when we are studying tumors, previously a doctor used to take a biopsy, a tumor slide, and look at the microscope and say that, look, this is a highly differentiated tumor, undifferentiated tumor, moderately differentiated, this is the cell type. And they used to make a lot of assessment to help the clinicians from there. So slowly it started coming out that really, this is not enough. Many highly differentiated tumors suddenly have a very bad outcome. The cells go rampant. And many undifferentiated tumors, well, at least some of them, don't do as badly as we think there is. So people and last few decades now, people have been dissecting out the molecular biology of these tumors. And having done that, they find that, even a tumor which looks so similar, 2-3 tumors looking so similar under the microscope,
actually are very different. They are different diseases. They are different because the molecular pathways that go into them, they are different. So essentially, if you look at a tumor in form of molecular disease, molecular undifferentiated grade 4 glioblastoma brain tumor, which used to look the same to everybody, is now essentially many types of tumors. So each one has its own outcome, each one has its own behavior, and this is something that I'd like to see. So I'll not really go into this introduction, but gliomas are different cells of, are the supporting cells of the brain, as you all know, in this. And obviously, when we've tried to, I don't think this will be seen, so it's okay. That's okay. So heterogeneous group of tumors, even cellularly. And then we can see, as you can see in the middle, WHO, we'll just call 2, 3, 4, because they are very simple types of tumors. Glioma grade 1 is a different kind of malignancy. So we'll not talk about it. Now you see what happens is, when you look at the histology, I don't think the point will work, but you can see from the left to the right, you can see the cellularity increasing, the differentiation getting less and less, more mitosis. And when the tumors grow very fast, they outrun the blood supply. So they are starved and the center dies. And so you have necrosis in the middle and new blood vessel formation. So last thing said microvascular proliferation. So tumors actually, nowadays antibodies to VEGF are one of the main, very important things in glioma. So recombinant antibodies of VEGF will also come about. So this is what you're dealing with. Now, what happens is that previously people would look at the tumor and say, okay, different grade 1, grade 2, grade 3, grade 4, depending on what they look like. Now, since recently, since 2016, WHO, now the pathologist will have to now take help of the molecular biologist in order to give his diagnosis. So this is what I want to emphasize to you, that suddenly our role has, of at least the people who are doing molecular biology of tumors, have shifted from research into actually frontline diagnosis. You know, when every surgeon will need a biopsy, and the biopsy will be taken, and from the biopsy you will have you will get a readout, and on that the doctor will prescribe chemotherapy or radiotherapy or surgery, whatever. But now, ideally speaking, though we are not yet prepared for that as a country, or as most countries are not prepared for this. Even USA has difficulty in, and Western Europe have difficulty in using it. But they integrated phenotypic and genotypic parameters. So in a tumor, you will give a readout. It's an undifferentiated grade 4 tumor with this mutation, P53 normal, that mutated, this one has amplification, and so on and so forth. And this will be the complete diagnosis. So what it does, as you can read from the slides, it adds objectivity. So suddenly you have classified a tumor as a molecular disease, and two tumors which look very same are essentially different tumors with different molecular readouts. So then what it does is, it gives you a biologically homogeneous group with a narrowly defined diagnostic criteria, and it gives you greater diagnostic accuracy. I mean, we have been doing this earlier. I don't know how many of you may know, but typically we have been looking at estrogen and progesterone receptors in breast cancers now for decades. That if it is a hormone positive tumor, hormone negative tumor. So that's one aspect, but it has now gone really into depth. And so you have better classification. Even clinical trials are now not on glioma grade 4. They are on glioma grade 4 with this molecular characteristic. because so the diagnostics and the therapy are merging together. This is where we are. So you can see the difference. Suddenly, if you see 2007 and 2016, if you compare, how do we have started? And now we have 2021, which is even more sophisticated, but this is the first time it came in. So here you have WHO grade 2 low grade infiltrative astrocytoma with cellular ATPR, unusual cells. Then you look at it and you can say. But in 2016, what are you supposed to say? You have to find out whether it is an IDH mutant or not, isocitrate dehydrogenase mutant or not, because IDH mutant tumors behave differently from IDH wild type tumors. Okay. So, and of course, at that time it was restricted to that. And you know, if you didn't have that, you could say NOS, not otherwise specified. Same way, so IDH mutant and wild type has gone into WHO grade 3, WHO grade 4, and so on and so forth. So here, so already now what has happened now, in 2021, there was this group called C-Impact now, Consortium to Inform Molecular and Practical Approaches to CNS Tumor Taxonomy. So you have gone into the pathologist domain, and the pathologist, and what it is done is shift to within tumor type and grading. So you know, and this is not happening only for brain tumors, it is happening for all tumors. So essentially, molecular biology is now on the forefront of diagnostics and treatment. So just to give you an example, it has no meaning in terms of, you know, or there's no need to work out what, but just to tell you how, in the sense how things have become more and more sophisticated. It has meaning. It has no immediate, I mean, I don't, you know, we're not going to go into the depth of everything, but just to give you a flavor of how things change. So in WHO, you know, we have tumors which are called astrocytomas, which arise from astrocytes. And of them, the most malignant grade 4 used to be called glioblastoma. It blasts out like a glial bomb. And there, in astrocytoma, you have what are you supposed to look at? You are supposed to report from your lab, your diagnostic lab, whether it is an IDH1 or an IDH2 mutant tumor. So you have to give a mutation status. Now, fortunately, it's easy sometimes to do it by immunohistology because the protein changes because of the mutation, and you can get actually mutation-specific antibody to do your immunohistology. Then ATRS is another gene. You look for that mutation or not. You ask yourself and you report whether P53 gene is mutated and this CDKN2A/B. Are they amplified or not? So many molecular readouts you have to give to that tumor in order to classify and tell whether it is grade 2, 3, or 4, in addition to what you are seeing. In glioblastoma, you can't have a glioblastoma. Previously, you know, grade 4 astrocytoma and glioblastoma are not very different. Today, you can't have that if you have an IDH mutation. So you have to have an IDH mutation to say, okay, so wild type to call it a glioblastoma. And along with it, of course, there's EGFR amplification, third tumor mutation, and so on. And not the purpose of this is not to give you a list. The purpose of this is for you to appreciate that today we are moved on. So this is a typical picture of a patient who had a brain tumor, as you can see from the slide, and the histological diagnosis favored a low-grade glioma because you know, it was, the cells were not that, let me say, mitotic or not that undifferentiated. But then you analyze molecular markers in the same patient and you find that it is a P53 mutant, it is an IDH mutant, and you did a FISH assay and you find that it is CDKN2A be amplified, as you can see from the multiple dots. I mean, no, if you can't see it, just believe me. In the slide, so that the final diagnosis is from grade 2, it actually has become grade 4. So that means the tumor is going to do worse than what you actually look at it and find out. So this is the importance of doing molecular markers. I'll not go into epidemiology and all that. And the treatment, of course, is surgery, radiation therapy, drugs. We haven't really gone much into it. You know, we haven't really been able to use our molecular knowledge to benefit the patient very much. Unfortunately, there's a huge amount of research work in that field. So this is something that we may need to look at. And the prognosis is of glioblastoma is still very poor. 1 year survival is 25%, 5 year survival is 5%. And there are these reasons. Another thing that I must tell you, just as we are chatting anecdotally, that you know, we need to see where the drug of choice is temozolomide. Now, temozolomide is a drug which adds a methyl group, and there is a demethylating enzyme, and that way MGMT, methyl transferase, methyl guanosine methyl transferase. And if that enzyme is high, then it counteracts the level of temozolomide. But if you have a promoter hypermethylation, which is suppressing the expression of temozolomide, then the drug is more active. So there's actually, even within a tumor, hypermethylation makes a difference. So this is something that we would like to think about, as how even in drug response, the molecular typing can make a difference. So therefore, more studies also. So what happens is we have done a little bit of a classification, but you know, things haven't stopped, things are still to improve. And what we have been doing in our lab is to look at one gene which is called the FAT1 gene, which has a role in making the glioma more progressive and invasive, and the knockdown reduces tumor growth and expression. So this is our research work. And in that sense, I'll just say that it's a member of the cadherin superfamily, and you can see that it participates in cell-cell interaction, and it's an atypical cadherin. So the molecule sticks out of the cell and interacts with the other cells, and there is a signaling pathway. And this is a very strange gene, which sometimes in tumors it's positive, is an oncogene, and in some tumors it's a tumor suppressor. And nobody really knows why a tumor is it's a tumor suppressor and why it is a positive thing. So, but in a Drosophila, people first found it out. Why is it called fat? Because it gives fat embryoid structures in a Drosophila when it is mutated and non-functional. So people thought initially it's a tumor suppressor. In our work, and even in many human cancers like breast and all, in it is in leukemias, it's an oncogene. In others, like some oral cancers, it's not an oncogene, it's a tumor suppressor. And we, in our lab, have been working on it. I think Dr. Kunzang, who is now professor in our department, did it during her MD and her PhD, and we've been continuing to work on it for the last almost 15 years or so. So we have elucidated the pathway, and we find that it actually generates pro-tumorigenic inflammation. So because of shortage of time, I'll not really go into depth, but what the take-home message is from my talk, which I think I'll truncate a bit because I've already done my 15 minutes, is that FAT1 overexpression promotes an immunosuppressive microenvironment. So it produces pro-tumorigenic inflammation in the cells, but it secretes, sorry, uh, I, I don't think we'll be able to see the data slide, so there's not much point in going into it. But then it essentially, we were looking at TGF-1 beta, TGF beta 1, TGF beta 2, and serpines, which is a TGF beta target in GDM samples. And what it does, it secretes these cytokines, and when you knock down the cells, then the cytokine release falls. As you can see, I don't think I can point out here, but you can see the levels falling, and at the protein and the RNA and the protein level, and also transmigration of monocytes, which we have been using in the THP model. So when you, what happens is this TGF beta, it repels monocyte infiltration. So it actually nowadays is the era of tumor immunotherapy. So we are looking actually for markers which will influence the efficacy of tumor immunotherapy. And we believe that this FAT1 is a marker which will, if you have high FAT1, you have high TGF beta secretion, which will repel the anti-tumor monocytes, and also this is regulated by a microRNA that we have shown. And most important is that it mediates the immunosuppression in glioma by another way, that myeloid-derived suppressor cells, these actually are attracted towards the tumor. So in a way, the tumor is behaving like a independent living organisms, trying to create conditions and evolve in a way that it repels the immunotherapeutic response of the body. So essentially, I don't know whether you can see, but there's a clear-cut difference in the monocyte migration. If you actually have a high-fat secreting cell, or high-fat expressing cell, not secreting cell, expressing cell, the monocyte migration is low. You knock down by siRNA and the monocyte migration becomes increased. So we have also now developed CRISPR knockouts where we will be able to study this phenomenon in long term as well. So therefore, the take-home lesson is that you have a pro-tumorigenic inflammation in tumor cells because of FAT1 overexpression in normoxia. And the other thing I didn't talk about is hypoxia. In hypoxia, even there's more increase and there's more HIF1 and more inflammation. In glioma, we have studied in detail, but it's possibly true in other tumor types. But then it also generates an immunosuppressive microenvironment, and it inhibits the infiltration of cells with anti-tumor activity and promotes it for cells with suppressive functions. And so there's a combined pro-tumorigenic effect. So we somewhere maybe later, if our work is intense and more rigorous, along with other markers, it's a possible marker for again looking at your tumor behavior. So this is our collaborators, Dr. Kunzan, who's been working on it since her MD thesis and PhD, and now continues in the lab. Partho Chattopadhyay, both are professors in the department, and also our Dr. Chitra Sarkar from pathology, Dr. Mahapatra, Sharad, Ashish, and Deepak. And the work that I had been briefly showing about the immunology is the work of Dr. Fulpul Irshad, who is there in our lab, is a very senior research scientist and a full bright scholar. Thank you very much for this opportunity. Thank you.
Thank you, sir, for the enriching insight and being the source of light, guiding us all on the path of glory. I would now like to invite Professor M. Afshar Alam, sir, Vice Chancellor Jamia Hamdard, for the presidential remarks. Professor Mohammad Afshar Alam completed his post-graduation in computer application from Aligarh Muslim University and PhD from Jamia Millia Islamia. Presently, he is working as Vice Chancellor and Professor and Dean of School of Engineering, Sciences and Technology at Jamia Hamdard. His research areas include software re-engineering, data mining, bioinformatics, fuzzy databases, and sustainable development. He is conferred with many prestigious awards like Bharat Samaj Ratna Award, AMP Award for Excellence in Education, Cooperative Citizen Award, World Environment Day Award, and Spardha Shri Award. He is also the member of various government bodies at both national and international level, including University Grants Commission and All India Council of Technical Education, National Assessment and Accreditation Council, and Department of Science and Technology, BSc.
Professor Survendra Shela, the former director of NBRC, Professor P. Prakash, former PVC Hyderabad University, and the Secretary General of the Society. Professor Raisuddin Saab, Advisor Research and HOD Toxicology Department. Professor Sohail Parveen, the Imam Saab, and other teachers, faculty members, and students. Good morning. Jamia Hamdard, you all know, is a reputed university both in India and globally, was recognized by the MHRD as a institution of eminence. We are in the process of going for their visit. Jamia Hamdard stands tall in NIRS university ranking, recognized, you all know, that #1 discipline of pharmacy. Recently, the Minister for Science and Technology, Sri Jitendra Singh, visited Jamia Hamdard on the occasion of National Science Day. He inaugurated the crypto TEM facility, which is supported by GST Burst program. We have 9 fees programs, spanning 4 schools for the program of GST. We have also DBT builders, and yesterday we have also the Ayush Ministry COP approved by Government of India. The Society of Neurochemistry India is one of the leading society in the domain of neuroscience in India and is actively involved in promoting research. Neurological disorders in the country are increasing at the alarming rate. The symposium is aimed at highlighting the issue and challenges faced by the researchers in exploring brain disorder and their mechanism through rigorous discussion and talk, and provide a compact platform for brainstorming dynamic approach. I am pleased to know that the main objective of SNCI is to enrich the young athletic neuroscientists with insights of brain functioning from the cellular to the systematic level, and to provide them hands-on training which will help to embed an academic and technological and translational approach toward the neuroscience research. I'm sure that the student will expose to multidisciplinary approach to understand the most complex organ of the human body. As Professor Babu has already announced that we are going to start a global chapter of this, really I congratulate to the society for doing so, and this is my second meeting I'm attending with Professor Babu, that as Professor Sohail is also working very hard, and recently DST recognized his work, and it is already in the DST portal. So the event has bring together the scientists from the academy across the country to deliberate upon new tools, new approach, method in implementation of improved solutions relating to neurodegenerative disorder. And Professor Sinha has already given the detail about the how he has compared from 2007 to 16 the disorder. A lot of research are coming up. So these are the wonderful symposium where students can learn some very many things about the brain research, because edema is a really big problem for now, and India is suffering a lot of this type of problem. So it is a very impressive various awareness platform and conference organized regularly to improve the exposure to the same and attract finding scientists and researchers in the field of neuroscience to cater to growing need for groundbreaking research in understanding the molecular and biochemical mechanisms responsible for this disease and discovering newer treatment modalities for the benefit of human welfare. Even I suggest Rais Saab and Professor Sohail, why not you create a center for some kind of neuroscience research so that that particular center can add many things. Maybe start from the toxicology department and other you can give autonomy to that center. Like the biotechnology have one more center is working. So in this direction you can think over this. And it's my pleasure to welcome all the delegates to the symposium on the neuroscience being organized by the Department of Toxicology at Jamia Hamdard in collaboration with the Society of Neurochemistry India. Thank you very much.
A seed signifies the immense potential of life within. A sapling demonstrates the initiation of the journey. A plant stands symbolizing growth. We at Jamia Hamdard aim for holistic growth and development of every individual's life. As a token for the same, we would like to present our dignitaries with plant a memento to signify the warmth of our bond and a token of our gratitude. I would now request Professor Prakash Babu sir to present a plant and a memento to our Honorable Chief Guest Sir Dr. Subrata Sinha. I would now request Chief Guest Dr. Subrata Sinha sir to present a plant and a memento to our Honorable PC Sir Professor Akshar Ali. I would now request Honorable Chief Guest Sir to present a plant and a memento to Professor Prakash Babu sir. I would now request Honorable PC Akshar Ali sir to present a plant and a memento to Professor SNCI Symposium sir. Thank you, sir. I would now invite Professor Sunil Parvesh sir, Organizing Secretary, SNCI Symposium, to come for the vote of thanks.
Bismillahirrahmanirrahim. Assalamu alaikum and a very good morning, everybody. It's a privilege and honor for me again to stand here and be part of an SNCI event. I start my vote of thanks with our Honorable Vice Chancellor, Professor Akshar Ali. When I came up with an idea only 2 weeks back, he was very encouraging. Even though exams are going on, most of the teachers and students are busy with their exams, but still to see more than 100 people here, 100 delegates of the conference, it's really my honor. I would also like to thank our chief guest, Professor Subrata Sinha. The moment I called him, he immediately agreed to come here, and he also himself offered that I would like to speak on my research, and that is so encouraging for us that even at this age and even at this stage, he is so much actively involved in research, and that should be an inspiration for all of us. I would like to thank Professor P. Prakash Babu, the guest of honor today. He is in the University of Hyderabad. He has been running the Society for Neurochemistry of India for many years now. I should convey thanks to him not only for all his roles in neuroscience, but the support that he has given to my students. From 2010, each year, one of my students is attending the workshops and the conferences organized under the SNCI banner. And today, most of them are abroad. And the SNCI Jamia Hamdard Neuroscience chapter in USA has been created by students of departments of toxicology who are faculty members, who are research scholars here. So it's really a pleasure. I would like to also thank all the teachers who are present here, all the delegates who have come from different parts of our country. It's a preamble to the DST Stuti workshop that will be officially inaugurated tomorrow. 30 workshop participants are here among us, and they have come from wide corners. From the south, we have from JSS College Pharmacy in Mysore. From the east, we have Alia University in Kolkata and Central University of South Bihar. From the west, we have the Central Institute of Medical Sciences in Nagpur. And all other places also, we have so many delegates. So that is really very encouraging. And I'm very thankful to all the faculty members, the scholars of our department, and also of Jamia Hamdard, who have done this immense job of organizing this within the past 10 days. I thank the Office of the Registrar, Maintenance, and everyone who came today and make this program a grand success. So thank you all. Thank you very much for coming.
I request you all to please stand up for the national anthem. Thank you all for your precious time. Kindly join us for high tea. All symposium participants are requested to immediately go at the registration desk for the registration and join us again post tea session at hall 1 for preliminary talks.
क्या? जी ना। ये तो चल रहा है। अरे वो कंप्यूटर सेंटर पर था।
हमेशा कौन सी वाली? ठीक ठीक। मैम दिखेगी, थोड़ी कुछ सेकंड लेते हैं। अभी बंद कर दो बाद में। जी
HDMI है तो हो जाएगा, HDMI से साउंड भी आ जाएगा ना।
सर साउंड पे अलग से लगा देंगे।
HDMI में शायद साउंड आ
होता है
जाएगा।
लेकिन सर इसमें आएगा।
अच्छा उसमें आएगा, ओके।
इसके प्रो में फिर उसके लिए
हाँ, तो स्पीकर में भी जाएगा। जब बुलाएंगे तब हम आएंगे। अभी पहले हमारे सुशील भाई को दे दीजिए, उसके बाद सेकंड में मेरा काला वाला में
सर का पीपीटी
थोड़ी सा हमको आप दिखा दीजिएगा, सो दैट कि हम चेक कर लें कि सब कुछ ठीक है कि नहीं। हाँ ये वाला SNCI
तो करना सर
हाँ डेस्कटॉप पे रख लीजिए और थोड़ा सा हमको एक वर्ष को चला के दिखा दीजिए बस। यहाँ तो नहीं आएगा, नहीं वहाँ नहीं आएगा, बस फर्स्ट क्लास। बहुत अच्छा लग रहा है सुशील भाई आपको देख के। आपसे वहाँ भी मिले थे JNU में, वो मंडल जी का जो बच्चा का था JRF से सारे। हाँ 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 आपका कमेंट मुझे याद है मैंने जी।
इट वाज नास्टी तो
नहीं नहीं नॉट नास्टी। आवाज नहीं आ रही। आगे आगे
ठीक है सर यहीं से इतने वास्ते से बड़े स्पीकर में पता
नहीं नहीं ठीक मिलेगा, ठीक है। आप इसको सेव करके इसको दे दीजिए। आप हमको दोबारा फिर नहीं आए क्या? JNU आए थे, आप बुलाते ही नहीं हैं। चलिए अभी देखते हैं कौन सा बड़े स्पीकर में करना है। ये आवाज होनी चाहिए इससे।
एक बार चेक कर लीजिए इसमें ठीक है। बड़े स्पीकर में तो सेटिंग में पड़ेंगे होती है।
आपका पॉइंटर की है
जी सर पॉइंट
किधर है?
सर एक बार चेक करिएगा साउंड अब चेक करिए। बड़े स्पीकर ठीक है सर।
आवाज कैसा आ रहा है?
छोटे पे रहने
छोटे भी
इन सर वो बड़े बड़े हैं।
बड़े बड़े तो है लेकिन आवाज इतनी
तो मैं कम कर दूंगा सर आवाज।
कम कर दीजिए। हाँ ये
ठीक है
बंद कर देते।
So I welcome you all. Now the sessions are starting, and it's really good to see young neuroscience students as well as scholars as well as faculty members in action. We have very focused 3 sessions. The first session is focused on 3 particular topics covered by Professor Sushil Jha from JNU, Professor Suman Jain from AIMS, and Professor Praveen Kumar from Delhi Technical University. They are well-known researchers in their own field, so I'm really happy that they have agreed to come. The second session will be the poster session at 1 o'clock. So I request you all, the moment we finish this, you all move to the first floor. The third session is based on funding opportunities and challenges, both for funding as well as how exactly you should get a patent in neuroscience. And the third, for encouraging all the young investigators, we have a young investigator session. So I request now Hans Prabha to introduce the chairs for the first session.
Good morning, I welcome you all to the first scientific training session. The chair was for the first scientific training sessions. Professor is the compound and Dr. Praveen Kumar in Delhi. So I welcome Dr. Shakespeare and Dr. Praveen stage. The main session. Sir, I would please start the session. Thank you.
Thank you. Now, so earlier, Delhi is quite a very renowned. And years. So, basically, so I did this to introduce and he's the way that his PhD was from the same. So so they invited the faculty, and then he was awarded many awards. One thing is very focused on the area of sleep and wakefulness. Only focus on that. Then only. This is also important subject to be know what you are. Even when you are staying and doing very so from one day, you don't need then he was thinking in 2005, and we got training merit-based level award again research provided, and research in the area of sleep, memory, creativity. So he is also a member of the editorial board of science. That's a function or divide for very so very important.
Thank you so much for such a nice introduction. I really appreciate it. Naisuddin has remembered my every event. And I really owe a lot of regards. So thank you so much. Thank you, Prakash Babu. You are actively organizing this kind of thing. And this offline meeting, I was just talking to Dr. Jain, but it seems that life is coming back. For scientists without conference, it looks like we are all not that active. So thank you so much. Thank you, Professor Pravesh and organizing committee for giving this chance to interact with you. And I delivered this lecture just to create awareness. Not what I do in the lab, I present the data, but generally I go with creating awareness what sleep does. So I do not know how many of you have liked this title, because fashion, being sleep deprived, if I say you are sleep deprived, no one would say that I'm sleep deprived. But are you sleeping less or compromising your sleep by doing every other thing and not sleeping properly? I think majority would say, yeah, I do that. So should we be doing that kind of or living that kind of lifestyle? That is my question. So if we do, is it a fashion, or are we inviting some disorders, especially cognitive or neurophysiological disorders? So if you ask me how much we should be sleeping, my simple answer would be 8 hours. Is it possible? In nowadays time, it is not possible. So how are you going to achieve it? There are several mechanisms. If the baby, like 1 year, 2 years old baby, if he or she wants to sleep, mother sings lullaby or some kind of tapping. Why do we do that? Why baby demands that? Means nature has given this power that there are mechanisms you have to adopt and you have to complete the sleep. That is the demand of the body. How much? It is just like one gets satisfied with 2 chapatis and the other gets 6 chapatis. So it is all the demand of the body, how much your body demands. You have to fulfill that. But one by one, I will go. Sleep, there are 2 stages primarily, non-REM sleep and REM sleep. And how do we study? We record EEG, brainwave, and what you see, the top one, when the EEG is desynchronized, high frequency wave, and lot of muscle activity, this is wave. And when EEG changes to high amplitude, low frequency, it becomes like low muscle tone, non-REM sleep. And very similar to wave, but then the muscle atonia, muscle doesn't work. Postural muscle doesn't work. That is a REM sleep state. And it's called paradoxical sleep, because we go in a comatose condition every night for few minutes to few hours. So I'm not going to talk about the REM sleep. It's very interesting because most of the dreams come during REM sleep. Now, if we say what sleep does, there are a lot of functions, and today we'll talk about cognitive, how it helps in our cognitive ability, and what are the problems that those are associated with sleep, that is called sleep disorders. So I will show some video. This is called fear conditioning. In Hindi, we say, "Doodh ka jala, chhach bhi phuk phuk ke peeta hai." Everyone must have heard it. What does it mean? Once you are scared, you are not going to do the same act. Or if, say, some house is a haunted house, ghost is there, no one wants to go into that. How do we learn that? So there are several examples. In the animal model, this is called fear conditioning. So what we do, we put the animal in the context and we give electrical shock, mild electrical shock. Next day, we bring the animal in the same context, no electrical shock, and animal shows freezing response because animal remembers yesterday electrical shock was delivered to the animal. You will see this video. When animal will jump, that shock is going over there. So it's a normal pattern, mild. And we give this shock for a second, not more than seconds. So when you see the animal is jumping, since the shock is being delivered to the bee, and we give it for 3 times only. That's the shock period. And next day, when same animal was brought in the same environment, you see how animal will start moving, showing fearful response. And computer registered all those things. This is not manually done, it's a computer registered program. And if the movement is not going more than a few seconds or something, computer registered as freezing. So that's the common phenomenon. Even if we are scared, we show freezing response. We don't move. So animal shows freezing response that we registered. And then what we do after training, after this shock, we put the animal into 3 categories: sleep deprived, stress control, and non-sleep deprived. You see, non-sleep deprived and stress control animal shows a lot of freezing, but sleep deprived animals did not learn this freezing response. Means they did not find this environment is scary. Next day when we test it. And sleep deprivation, 6-hour manual sleep deprivation, soon after. That is the beauty of this experiment. Another, this is called appetitive conditioning, means we give animal fruit to animal fruit juice, and that is in a sequence wise we give that fruit juice. Because that was electrical shock, one would say that because of the scary phenomena, the cognitive ability maybe get hampered. This is the positive response, positive reinforcement. Allow fruit juice. And fruit juice we give in a sequence wise. First sequence is and the fruit juice will be delivered from this chamber. Outside, a lever, there's a window here. This is second phase when the light is off. The third phase when the fruit juice will come and see animal is ready to take fruit juice. And if animal doesn't, animal is not alert, or doesn't pay attention, then the liver will be down. So animal has to be alert, need to learn what time tentatively the juice will be coming. And the same experiment we perform, and what you see, sleep deprived animal is showing impairment. They did not learn, they did not judge properly precise timing when the fruit juice will be coming. So that shows 6 hours of sleep deprivation soon after training is causing memory deficit. The animal who learned properly and slept properly showed good response, but the sleep deprived one did not. What does the student do? They learn, especially during examination time. Keep mugging, whole night awake, sleeping less. So just see, imagine how much effort you are putting in if you are not sleeping properly. How much you are going to remember. Now the question is, how does sleep deprivation affect memory consolidation? We performed certain molecular experiments, like some candidate genes. These are FMN2, CSK3 alpha and CT and SHANK3. These are tentative memory candidate genes. And what we did was establish whether these genes are. So we saw the expression level of these genes are very high soon after learning. And then when we use siRNA to inhibit the expression of these genes, so this is what you see, this freezing response in the animal. So the control one showed good amount of freezing, and the knockdown one did not show good amount of freezing after training. So that shows these genes are the possible candidate, memory candidate gene. And when we sleep deprive them for 6 hours, and when we estimated their expression level, you can see this the sleep deprived animal showed less amount of expression of these genes. So what does it mean? That sleep somehow potentiates the expression of these genes, helps in the expression of these genes. In turn, it helps in memory control. If you are not sleeping properly, some of these genes are not getting expressed properly. Now, another candidate is, this is called adult neogenesis. In our brain, number of neurons are set. After a certain age, 4 or 5 years of age, neurons don't divide and number remains fixed. If those neurons are damaged, they cannot be recovered. Most of the neurological disorders are related to the neuronal damage. But two areas, till date what we know, the two areas in the brain, adult neurogenesis are taking place. One in the dentate gyrus of hippocampus and other in the subventricular zone. And hippocampus is a very important area, especially in terms of learning. So most of the learning where hippocampus is involved. And it has been found that neurogenesis helps in memory consolidation. So what you see here, a very nice demarcated developing neuron in the dentate gyrus. This is learning. When we give the task, animal learn properly, and then there are mechanisms. You inject BRDU because the BRDU is an analog of nitrogen base. It gets incorporated in DNA. So all the dividing cells will have a BRDU. Then you express antibody against BRDU, and you will get the BRDU positive cells, and that will show these are the newly divided cells. And then there are measurable markers show developing neurons. So what you see here is DCX. These are BRD and DCX positive cells, and show that how nicely these neurons are developing and generating their projections in the dentate gyrus. But in the learned animal, and if they are sleep deprived, here we have to sleep deprive them for 24 hours, that is REM sleep deprivation. Why 24 hours? Because cell division takes 16 hours to complete. So unless you provide that 16 hours, cell will not divide. So we have kept the animal as a mechanism to put them on platform where their REM sleep will be deprived. Non-REM sleep will be intact. So these are REM sleep deprived animal, and you can see in REM sleep deprived animal, everything is broken. No processing. All these neurons, what you see, the swollen neurons, they have not developed the projection properly. So, and these are the dividing cells that is in terms of like when we learn, that triggers this division. And it has been shown that these divided cells remain in the system where they keep this information for a longer period of time. So memory retrieval, these divided cells, newly converted cells are playing important role. But if we learn and then remain sleep deprived, they are not going to get formed. So obviously there will be cognitive deficit if someone is not sleeping properly. This is a very important because this is also a kind of workshop, and this is very close to my heart. What happens, the another mechanism, what sleep does, I do not know where you will be, you have read some paper or not. What we learn when we are acquiring information, there is a different way of the pattern of neurons, they get fire, like action potential is generated, especially in the area those are involved with the memory retention in the dentate gyrus, amygdala, all those areas. And when you sleep, those neurons are similarly getting reactivated. That has been shown, reactivation has been shown, and this is called synaptic re-entry reinforcement hypothesis. And they have shown that how those neurons, those are involved in the acquisition, are getting reactivated when someone is sleeping. And if you are not sleeping, this will not get reactivated. And reactivation is the same thing for the neuron. In common terms are what I say, when you are reading something, trying to mug up something, what we do, we keep repeating. So more you repeat, more you can memorize. So more reactivation of the neuron that is taking place during sleep means it is facilitating the memory consolidation. But the question is, and these are my words when I was in Philadelphia, what we can see here, reactivated neurons during sleep, when we generated that kind of synaptic plasticity. That is some kind of tough experiment. I'm not going to explain that. But what we did here, the question is, when we are sleeping, when the neuron gets generating action potential, it cannot generate action potential unless there is a stimulus. So when we are learning, the stimulus is there. And it is easy to understand that those neurons will be generating action potential. But when someone is sleeping, no stimulus, how those neurons are getting reactivated, from where the stimulus is coming. So this is a reverberatory mechanism in the brain. And that happens because when we sleep, what I showed EEG, they have different waves that brain generates. Alpha, beta, delta, gamma, theta. Theta is more prevalent when we go into REM. Delta is more prevalent when we are in non-REM sleep. And there are other circuitries as well, like PDO waves. So I call it the jumping waves. They jump. They're generated from pontine area. From there, they jump to the thalamic area. From there, it goes to the occipital cortical area. So those are the jumping waves. And that happens during REM sleep. We do not know why these jumping waves are there during REM sleep. But implication I'm just trying to understand, when we train the animal and they learn it properly, and when they are sleeping, you can see this delta wave and then some rhythmic firing is there in the brain wave. Theta wave is more prominent in the learned animal. And these waves are generated specifically during sleep. And these waves has that kind of energy that helps in the neuronal communication. So this provides stimulus to the neuron to fire action potential. So theta waves, delta waves, the energy level goes up. When they cross certain level of energy, they trigger those neurons to replay the firing pattern the way they were firing during acquisition. All are connected mechanisms. And when you do not provide that stage to the brain, how they are going to perform their task. So cognitive deficit is quite logical. That is what my take here is. And some of the interesting things that I would like to show here. If you see, this mother duck is sleeping because tuck-in pattern is the sleeping pattern in birth. But what you see, the eye, this side eye is open. And their babies are sleeping. What does it mean? It means nature has also given some evolutionary power to sleep. Birds and especially aquatic mammals, because aquatic mammals have to breathe every time. So they can't sleep. If they sleep, they will sink. And how to gulp air. So one half of the brain sleep, other half remains active. And this is what this bird is doing. The another side of the cortex is active. This side is open, watching the environment if there is no threat, and the mother is sleeping. So this is called brain lateralization. One side is sleeping, another side remains active. Dolphin does the same thing. So nature has never said don't sleep. Given the mechanism how to sleep, adapt that such a way that you sleep. And that challenge is for human being nowadays. Because in this competitive world, everyone has to work. They have to perform their job. IT professionals, whole night they have to be awake to do their job. So how to develop mechanisms that we have to see, because others are showing us there are mechanisms. Common disorders, I'll just show some videos. You may be knowing insomnia, hypertonia, apnea. These are the common sleep problems. But there are some interesting sleep problems like periodic leg movement syndrome. Most of you must have seen grandmother, mother sleeping and jerking their leg, and it's very painful. But we do not know it's a sleep problem. Another is narcolepsy with cataplexy. I'll show that video. Sleep-related eating disorder. The patient who is suffering from this eating disorder doesn't know what he or she does. Sleeping, will get up, go to the kitchen, eat something, come back, sleep. And if you ask, do you remember what you did? No, I don't know. No remember. REM sleep behavior disorder, you will see all those videos. So this is periodic leg movement syndrome. Every 2 minutes there is a periodic cycle, 20 seconds. And it's a very painful situation. What is prevalence? Prevalence is 4 to 11% in adult, 5 to 8% in child. Old age, forget it. There will be more prevalent. And what happens? And what happens? One cannot sleep properly. And it has been shown, especially in child, that this is hyperreactivity, delayed language development. The child who is suffering from this periodic leg movement syndrome, poor concentration at work in adult. So these are the consequences of these disorders. Another is narcolepsy with cataplexy. So what happens? REM sleep, I said, muscle tone is not maintained. Postural muscle, breathing muscle work. So if someone is going into REM sleep from wake, it is not possible. So you have to go from wake to non-REM, then REM sleep. Otherwise, muscle atonia in REM sleep would cause problems. If I am talking and I can go to REM sleep, I will be flat on the floor. But in narcolepsy, probably it happens. That subject goes from wake to REM sleep-like state. And this is what you can see what happens to this lady. She's doing some exercise. Flat on the floor. Trying to wake up. Again, sleep attack. Very difficult to maintain proper concentration. This is, I'll show the whole video, sleep-related eating disorder. Oh. What happened? There's a problem in the audio, I think. No reconnection. That was sleep-related eating disorder. I do not know what is the prevalence of this disease in India because we do not have any data. We do not have any data because it doesn't come out because this looks like a disgusting kind of phenomenon. But patient subject should come out. We should be knowing. But in US, this is 1 to 2% in age. REM sleep behavior disorder, what I said, muscle tone doesn't work, muscle is atonic. But when REM sleep behavior disorder happens, there is a state called REM sleep without muscle atonia. Means what you dream, what you think, you start acting like that. So here you can see this gentleman, what he must be dreaming something, and then all of a sudden you will see hand movement or something, he's trying to grab something. Time is short, so I'm just rushing. This gentleman is talking, laughing, and making you cry. And these are the videos from hospital because their EEG is getting recorded. And the most dangerous part is this one. Because this gentleman is sleeping and all of a sudden he thought that he's trying to crash someone, so start beating. So you will see in that video. And if you do the Google search, REM sleep behavior disorder, you will find lot of bed partners have been hurt by their partner if they are suffering from badly. Just Suman बैठी है Suman will tell क्या है हकीकत है या नहीं. No, actually, just imagine that there is child, grandchild. Generally, what we do, like, चलो भाई बाबा के साथ, दादा के साथ, नाना के साथ सो जाओ. If Baba Dada is suffering from, just imagine if the child is sleeping, what would be the condition of that child? Okay, so these are the real sleep-related disorders. So with that, I just say thank you. Thank you for giving this opportunity. These are my lab heroes. They do all this work over here. So thank you. Okay. Actually, that girl she has raised her hand first.
So I would request Professor SNCI Symposium, Advisor Research and Head Department of Toxicology, to present a memento to Professor Sushil Jha. I request Sir to present a memento to Professor Suman Jain. I now request Sir to present a memento to Professor Praveer Kumar. Thank you all for the patient listening. Kindly join us for the lunch, and all participants are requested to display their poster on first floor in poster presentation arena. I request you all to join us again for plenary session 2 post lunch. Thank you.
आइए जैसे लंच कर लेते हैं। आप ये आपका ले ये अच्छा पहले से रखें। हाँ 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 मिल गया बिल्कुल मिल गया। नहीं
आज जो नेक्स्ट स्पीकर्स भी हैं उनका भी लगा दो। गौरव सर का क्या है? गौरव सर भी है क्या?